In this video, we will discuss the process of preparing a microscope slide for observing plant cells, specifically using onion epidermis. First, we begin by carefully peeling off a thin layer of the onion's outer skin. This delicate layer is then placed onto a clean glass slide. To ensure that the specimen remains hydrated and flat, a drop of water is added on top of it. Next, we take a cover slip and gently lower it over the specimen, which helps to flatten the layer for better viewing. To improve our ability to see the cellular structures, we will apply a stain, such as iodine, to the slide. This step enhances contrast and makes the components of the plant cells more visible. Once these preparations are complete, the slide is ready for observation under a microscope, allowing for a detailed examination of individual plant cells and their internal structures. We will also discuss the process of preparing a microscope slide for observing animal cells. First, a small piece of liver tissue is taken and crushed to create a thin film. This film is then placed onto a glass slide. To improve the visibility of the cells, a stain such as methylene blue or iodine is applied. This staining process helps highlight the cellular structures. Once stained, a cover slip is carefully placed over the specimen to flatten it. With this preparation complete, the slide is ready for examination under a microscope, allowing us to observe individual animal cells and their intricate structures. In microscopy, two key methods for preparing samples for viewing under a microscope are the transverse section and the longitudinal section. The transverse section involves slicing the structure perpendicular to its long axis, resulting in a cross-sectional view that reveals the internal architecture at a specific point, similar to cutting a hot dog bun in half horizontally. In contrast, the longitudinal section entails cutting along the long axis, providing a lengthwise perspective that shows the internal organization from one end to the other, akin to slicing the bun vertically. These sectioning techniques are vital across disciplines such as biology, anatomy, and materials science, as they enable detailed examination of the internal structures of organisms, tissues, or materials, thereby enhancing our understanding and analysis of complex structures. When we look at the transverse and longitudinal sections of a plant stem, we can learn a lot about its structure. In the transverse section, which is cut across the stem, we see a round shape with different layers. The epidermis is the outer layer of cells, the cortex is a layer of cells that help store and support the plant, the vascular bundles act like veins, with xylem for carrying water and phloem for transporting nutrients, and the pith is the central part made of similar cells. In the longitudinal section, cut lengthwise along the stem, we can see the whole length, showing internodes, which are the sections between the points where leaves and branches grow, called nodes. We also see the vascular tissue, where the xylem and phloem run along the stem. Together, these sections help us understand how the plant is built and how it moves water and nutrients around. When examining magnified views of animal and plant cells under a microscope, their appearances differ significantly. Starting with the animal cell, it generally exhibits a rounded or irregular shape, with a prominent nucleus often positioned centrally. A key feature of animal cells is the absence of visible cell walls. In contrast, plant cells tend to have a more rectangular or box-like shape, attributed to their rigid cell walls. Although the nucleus is present in plant cells, it may not be as prominent as in animal cells, and the surrounding cell walls are clearly visible. It's essential to note that these characteristics are general. The specific appearance can vary depending on the cell type and the staining techniques used.